All right. Good morning, everyone. Can you hear me?
Yeah.
Okay. So the first thing that I want to mention is that I have sent everyone the instructions for the course assignment. So I'm not sure if you're able to see this. So I sent this layout of what the course assignment is going to be. I will, as I mentioned previously by email, I will record just an overview of information that's going to be necessary for you to have the context. But basically, please review these instructions as it provides you what is the actual project itself. It gives you the layout of the format, in addition to giving you a set of deadlines that corresponds to different submissions along the way to develop this actual course project. It will be an individual project that I will assemble together as a group project. It will involve both a written component and also a presentation component. The presentation component will be discussed a little bit later. We will have like dates that are available so that everyone will present their respective projects. So again, I will have a recording that specifically denotes all the details with regards to this project, but I wanted to make this available to you as soon as possible because there are pending deadlines that are coming soon with regards to selection of topics. Okay, so if you have any questions or doubts after I provide you with the recording, just let me know so that we can clarify them. Okay, so let's go ahead and get started with the lecture. Okay, so this lecture is longer, so I won't be able to get through all the slides. So some of this will be carried over into the recording. But basically, what I wanted to do was to, just like we did with proteins, was to give you a bit of an overview of nucleic acids, but also dive a little bit into the topics of transcription translation in terms of techniques, as subsequent discussions in the course will touch upon techniques that manipulate these pathways in order to, provide chemical biological tools that give us insight into important biological pathways. And so let's go ahead and get started. So in our previous lecture, we learned, or previous set of lectures, we learned from Dr. Tommaso Cupido about the design of small molecule probes to observe and copy nature and to perform de novo biochemical phenotypic screenings, which led to our Journal Club 2, which really specifically focused on a chemical genetic strategy to develop protein mutants for small molecule inhibitor synthesis, and the idea was to obtain insight into the functions of AAA proteins. Through this journal club, we learned a bit about differential scanning fluorimetry and isothermal titration calorimetry for biophysical characterization of the protein and small molecule interactions. Those were the key techniques that I wanted people to really zoom in on as far as they provide insight into, quantitative interactions, giving you some amount of stoichiometry, but also telling you about how things either become stabilized or destabilized in the presence of, let's say, in this context, small molecule interactions. All right, so as we discussed in the beginning of the course, when we're thinking a bit about the fundamental biology of life, and we're talking about the processes that, are inherent to what allows us to live. And we're talking about DNA transcription to RNA and then RNA translation to protein. And we did talk about some briefly, some of the pathways that can lead to one to another, but essentially this movement of information from DNA to proteins and get a perspective of where this is occurring within the cellular environment. So to discuss this, though, it's important that we have a review of nucleic acids, as you all have seen nucleic acids either in your biology courses or in your biochemistry courses. So a little bit about the building blocks of nucleic acids, which are the nucleotides. Okay, so in general, just a perspective of their function, they do participate in energy exchange processes and metabolism. One of the key molecules is adenosine triphosphate, more commonly known as ATP. They can serve as metabolic intermediates. They participate in cellular response to extracellular stimuli and intracellular signal transduction, CAMP. They participate as structural components of enzyme and enzyme cofactors such as NAD. The constituents of nucleic acids, they are the building blocks, the molecular repositories of genetic information. So principally, we're talking about DNA and RNA. As far as the structural composition, so there are key three components to keep in mind. So you have the nitrogen base containing bases, so the nucleobases. They can be in the form of purines or pyrimidine. Then you have the sugar component. In this case, we're looking at pentose. And then you see here the at the position carbon 2 prime, where you have it highlighted in red, but also in black, depending on which form can be the OH form or the H form. And the other component is the phosphate group. So let's talk a little bit about the nucleobases. So they come in the two main forms of pyrimidine and the purine. So these are nitrogen-containing heteroaromatic molecules. They allow for planarity in terms of the structure, and it actually becomes a big component in terms of the how nucleic acids, have adopt a certain structure. And a very important component of them is that they absorb UV light in the range of 250 to 270 nanometers, and that's actually quite important in terms of spectroscopy, because the typical way that you can get a relative measure of the amount of nucleic acid is by the absorbance at 260, and that's coming from these bases. And so that's typically a way that people can measure absorbance and correspond it to a relative concentration. So these are representatives of the pyrimidine bases, so cytosine, thymine, uracil. And so you see that there are some that are commonly seen observed in DNA and those that are for RNA structures. So cytosine is found in both DNA and RNA, thymine found only in DNA, uracil found only in RNA. And a critical component about these structures, so we mentioned the planarity, but also they serve as very good hydrogen bond donors and acceptors. And so that forms a big component in terms how these contribute to the formation of nucleic acid structure. You have the corresponding pK values of, and here's for cytosine at the end 3, 4.5, thymine, N3 at 9.5. And so typically what you're observing is that these molecules are neutral at a pH of 7. Here are the representative purine bases. So you have adenine to the left, guanine to the right. And so these are found both in RNA and DNA. And similar to what I said about the other nucleobases, are good hydrogen bond donors and acceptors. Here are the corresponding pKa values. And also very importantly, they're neutral molecules when you have a pH of 7. Okay, so as I mentioned to you, a really important contribution to the spectroscopy of nucleic acids is this absorbance. So just a visual depiction of the absorbance of the different nucleotides. And so here you have the corresponding extinction coefficients of these individual nucleotides. And generally, what people will do is take the absorbance at 260 for a nucleic acid as a measure of that of those molecules. All right, so in addition to the nucleobases, we have the actual sugar ring. And so what you basically have here is the carbon numbers are given as prime designation to distinguish them from the numbered atoms and the nitrogenous bases, the nucleobases. And so they can come in two forms. And so here you have the representation of the structure of these sugars. So you have what is the OH form and then the H form, which is typically referred to as the deoxy form. Okay, so depending on like the how they incorporate in the structure, you have one or the other. Okay, so you have the OH form present in RNA and the deoxy form present in DNA. All right, so as far as the how this is within the structure of the nucleotides, essentially you have a beta and glycosidic bond. So the nucleotides, the pentose rings attached to the nucleobase via this type of bond. And so the connection in pyrimidines would be at the position N1, and the connection for the purines is at the position N9. So another critical component is the phosphate group. And so here, typically, when you're looking at the with respect to the sugar component, what you would typically see the positioning of the phosphate group would be at the 5' carbon. So in this carbon right here. So this would be 1', 2', 3', 4'. But you can also have modifications where the phosphate group can be at other positions. Okay, so these are just representations of other positions, such as being at the 3' position, 2', a mixture of the both. Okay, that's just to give us a perspective of how that can actually be incorporated. And so when we're looking at them in a more holistic fashion, just the composition of these nucleotides. So here we have the representation of the deoxyribonucleotides. Again, the critical component being that you have the H in this position. And so these are the corresponding names. In addition to the names, you have the characteristic symbols that are used. So you can have the one-letter symbol, you can have the three-letter symbol, and the denotation of the D for the fact that these are the deoxy form. And then these are the corresponding ribonucleotides. So these would not be the deoxy form. So you have the OH and this position here, the corresponding names, the corresponding symbols, either the one-letter representation or three-letter representation, and the names of them in their nucleoside form. So the nucleoside form would be without the presence of the phosphate groups. So, as we mentioned with regards to proteins and their building blocks, amino acids, there are 20 common amino acids, and then there are unusual forms. So that's also true for nucleic acids. So modification of nucleobases can occur, and that occurs after RNA DNA synthesis. As an example, you have 5-methylcytidine, common in eukaryotes, also found in bacteria. N6-methyladenosine, common in bacteria, but not so in the eukaryotes, and not found there. You can also have modifications, which serve as an epigenetic marker. So the function of these type of modifications is a way, and to an extent, can be a way to mark one's own DNA. So that marker indicates that the cells can degrade foreign DNA, and this can serve as a defense mechanism to distinguish one's own DNA from an invading pathogen. Way to mark which genes should be active. So that's another function of these type of modifications. And so here are representations of these unusual nucleobases, and just basically emphasizing the nucleobases that are attached to the sugar. All right, so those are the building blocks. So now let's go into nucleic acids. And so in general, they are biologically occurring polynucleotides in which the nucleotide residues are linked in a specific sequence by phosphodiester bonds. And so principal functions is that they participate in the storage of genetic information. So that's basically what we've discussed a bit about the molecular biology dogma, this translation of or this movement of information from DNA down to proteins. So again, the transmission of genetic information, that's regulated by messenger ribonucleic acid, or more commonly known as mRNA. You have the processing of genetic information done by enzymes referred to as ribozymes. And you have protein synthesis, and this is facilitated by transfer RNA and ribosomal RNA. So tRNA and rRNA. And another really important thing structurally is that these can be broken down into different levels of structure. You have the primary, which is essentially the nucleotide sequence of a nucleotide strand and its covalent structure. Then you have the secondary level, so that's any regular stable structure taken up by some or all of the nucleotides in the nucleic acid. And then you have the tertiary level, then that's the complex folding of large chromosomes within eukaryotic chromatin. So we're going to look a bit into these different levels of structure. All right, so let's first start off with the primary level. And so this is essentially the composition. So again, we looked at the building blocks, right? Where you have here are one of the nucleotides, the corresponding nucleobase, the sugar, and then the linkage at the 5' carbon to the phosphate group. Okay, so also you have a similar arrangement for the RNA group. Okay, key difference is what's positioned at the 2'. Okay, but now is what's linking one nucleotide to the other. And so essentially what's facilitating that is a phosphodiester linkage between in this case, this first 3' position to the 5' position of the neighboring nucleotide. And so it's very important that when we're reading these the sequence, right, because this is the primary level, the way that we read this is from the 5' end, and the 5' end is essentially where the phosphate group is connected to the 5' carbon of that nucleotide. And then we go all the way to the direction of the 3' end. And the 3' end is essentially where you're just looking at where this this group here has no actual phosphate connection. It's just this OH group here. So that is the typical pattern that you read a nucleic acid, the 5' end to the 3' end. Okay, so the successive nucleotides of both DNA and RNA are covalently linked through the phosphate group bridges, which the 5' phosphate group of one nucleotide unit is joined to the 3' hydroxy group of the next nucleotide. So the covalent backbones of nucleic acids consist of alternating phosphate and pentose residues. And so that's actually quite important because you think about the nature of these type of structures and how they will change at least some of the components of how these will interact. So you have these phosphate groups in contrast to the nucleobases. And so what does that backbone actually contribute? What that's going to contribute is a hydrophilic character. So that's going to be something that favors interaction with waters, right? You have these groups that are charged that are quite favorable for water molecules to surround them. The hydroxy groups of the sugar residues can form hydrogen bonds with water. The phosphate groups are completely ionized and negatively charged at a pH of 7. The nucleobases may be regarded in the perspective of the nucleotides as your, or at least nucleic acids as your side groups. All phosphodiester linkages have the same orientation, again, 5' to the 3' end. Now, it's very important to distinguish between DNA and RNA. In that DNA, the backbone is fairly stable. So hydrolysis accelerated by enzymes can lead to the breakdown. So much stable that you can have a half-life of DNA of 521 years. The DNA strands of a reasonable length could last, millions of years, and that's something that people have taken advantage of in terms of being able to characterize organisms that have been discovered over the years. Now, this is in contrast to the RNA backbone, which is relatively unstable. So in water, RNA lasts for a few years. In cells, mRNA is degraded in a few hours. It's something that's being constantly processed in terms of its regulatory roles that it plays. Right, so how do we represent the nucleotide sequence? And so I already mentioned that we go into from the 5' to the 3' direction. For a nucleic acid that consists of the AGCTA sequence, so we're looking at a particular sequence that consists of those nucleotides. So how do we represent them? Okay, so you can write it in the following format. For instance, you can refer to the 5' end by using this 5-letter sequence and putting a P at the start, okay? And then at the end, the A, you can then attach the OH so that you're indicating what's the start and what's the end. You can be a little bit more specific in terms of indicating the phosphate groups that exist between these nucleotides. But more traditionally, you have something like this, where you have the 5-letter sequence, where you indicate that the beginning is the 5' end and the end is the 3' end. But these are understood to be equal in terms of its representation of how you would define the nucleotide sequence. All right, so that's the primary level. Now let's move into the secondary level. Okay, so DNA molecules have distinctive base compositions. Kargoff and colleagues, this was in the late 1940s, found that the 4 nucleotide bases of DNA occur in different ratios in the DNAs of different organisms, and that the amounts of certain bases are closely related. So there was a connection seen between how certain nucleotides would interact with one another. So the base composition of DNA generally varies from one species to another. DNA isolated from different tissues of the same species have the same base composition. So what was observed, what is observed is in all cellular DNA, the number of the nucleotide A equals to the number of the nucleotide T. And so we're going to see in a moment why that is the case. I expect that you are familiar with this. Then the number of the nucleotide G will equal to the number of nucleotide C. And so that essentially what you're going to have is that the sum of the number of purines will equal to the sum of the number of pyrimidines. So I would say the classical structure that we're familiar with, and there are other structures, I'm actually not going to review them here for the purposes of time. Again, I want more the applications as far as like how we're going to think about this for chemical biology as opposed to like really deep dive into biochemistry. But nonetheless, it's important to review some of the features of this particular model, which is the Watson-Crick model. And so in this model, what you have is a right-handed double helix where you have your hydrophilic backbone. Again, that's the composition of the deoxyribose and phosphate groups. They face outwards, and so they're able to interact with water. So here's just a depiction of those groups that are facing outwards with respect to the double helix. Then you have the furanose ring of each deoxyribose in this C2 prime endo conformation. So I haven't really given you that perspective, but that's what that is. And then very importantly, what's facilitating the an important component of this type of helix is that you have the purine and pyrimidine bases, which are hydrophobic, and so by their nature, they're not going to interact with water. So in terms of how they're oriented, they're going to be oriented in a way that's away from the water. And so they stack inside the double helix and they're stabilized by hydrophobic interactions and are perpendicular to the long axis. So this is the long axis. And so what you see is this base pairing stacks that are perpendicular to how we define the long axis. Okay. So we're going to look a little closely at these base interactions to also see what's facilitating that structure. So we have the layering that's favored by these hydrophobic contacts or these aromatic contacts, one on top of the other. But there's another critical component that's helping to facilitate this arrangement. And that is that you have what is referred to as base pairing. So what is this? What's underlying this base pairing is essentially hydrogen bond contacts between these two adjacent, or I guess you would say neighboring nucleotides that's allowing them to have a very strong interaction. So base pairing occurs in which a nuclear base in one strand is paired in the same plane with a base on the other strand due to this hydrogen bond interactions. So you have that purines will pairs with a pyrimidine. So A will pair with T. So here we have this visual depiction of that, where we have adenine that's interacting with thymine, and the interaction is occurring by the hydrogen bonds that can be facilitated between the two. And what we observe here is this contact of two hydrogen bonds between them. Then you commonly will see the cytosine interact or excuse me, base pair with the guanine. And so here's the representation of this, where you have these three hydrogen bond contacts between the two. Okay, so the base pairing of the two strands creates a major groove and a minor groove on the surface of the duplex. And so that was better depicted in this picture over here, where you're looking along the long axis and you see this arrangement where you have this minor groove and this major groove that exists due to this base pairing interaction. So the DNA strands interact in an anti-parallel orientation. Okay, so what does that mean? So you essentially have two different strands that are forming these helices that are in contact with one another. And as I mentioned to you, when we typically read in the primary form, we read 5' to 3'. But the way these two strands are interacting with one another is interacting where you have one oriented in a manner that would be 5' to 3' end, and the other oriented 3' end to the 5' end. So you have this essentially this anti-parallel arrangement. Another important feature is that you have in terms of the stacking, the stacked bases are 3.4 angstroms apart. And each turn of the helix, when you're measuring from one minor groove to the next minor groove, okay, so you're looking at it from that perspective, will include 10.5 base pairs that are stacked. So here's just a representation of that, 10.5 base pairs that are stacked, going from one minor groove to the next minor groove. And in terms of the distance that's covered, there's covering a distance that's 36 angstroms. Okay, so continuing with secondary structure, but here we're moving from DNA to RNA. And again, I'm going to be a bit simplistic in terms of my description here. So RNA does not have a simple regular secondary structure. Generally, an observation is that you have a right-handed helix dominated by base stacking interactions, similar to what was just described for DNA. The base stacking is strongest between two purines. A pyridine between two purines is often displaced to enable the two purines to interact. The RNA can form a double helix. So RNA can base pair with complementary regions of either RNA or DNA. And so these are the common types of base pairings that you can see. The RNA double helix takes on the A-form right-handed double helix structure. I didn't actually um, go into the different types of structure again. This is something that would be more of a biochemical destruction perspective, and so I just wanted to give you an overview. What's really important is that as part of the way that RNA can form, you can have breaks in the A form helix that are caused by mismatched or unmatched bases in one or both strands that result in what would be more complex structures. And so, what these complexities are different features such as the introduction of bulges. So here we have this visual depiction, this cartoon representation of an RNA where you have this bulge that's formed by this A nucleotide that does not have a match. And so you have here this space that's being created, and because of that space, that basically gets, inserted out of that arrangement in order to more favorably have this interaction between these base paired stacks. You also have what are referred to as internal loops. So here's a representation of these internal loops, or a structure that's commonly referred to as a hairpin loop, where you have this type of segment here that basically then points outwards, and then you have this arrangement over here that is what's referred to as this hairpin loop. So this structure can become quite complex, again, due to these mismatches or unmatched bases. And now let's look at the tertiary level of structure. So here we're going back to DNA. So in terms of composition, you can have circular versus linear DNA. And so what really constitutes that? So DNA strands can exist in this double helical structures. Okay, so that's what constitutes secondary, but then how they can, become compact or arrange themselves. So you can either have circular, as is common in viruses, bacteria, and archaea. This form is also present in eukaryotic cells, or you can have the linear form, which is the dominant form in humans. Okay, so here's a depiction of circular DNA, to the right, the linear DNA. But as part of this, you can have then the DNA itself, this strand, become a supercoil. So what we have here is just a representation of this, where we have this double helix of DNA, that classical model. You have the long axis, but then it starts to coil up, and it coils up in such a way then you get this type of structure here, which basically is the strand, turning into itself. So cellular DNA is extremely compact because of this supercoiling process and requires a high degree of structural organization. The folding mechanism must not only pack the DNA, but also, very importantly, permit access to the information in the DNA. The compact structure is achieved by this supercoiling process. So here we have this depiction of then what happens to the supercoiling process, how that DNA becomes compacted. And so essentially, what you're seeing here is how the DNA is compacted in a eukaryotic chromosome. So the chromosome is a single large DNA molecule, and as part of the structure, you have its associated proteins. And so these chromosomes store and transmit genetic information. So what we're seeing here is this cartoon representation of how you go from the double helix, then the affiliation with um, these structures known as the histones, the associated proteins that help to take that double helix strand and basically wrap around it. And so you get this wrapping around it, forming these higher order structures, these supercoils, and then how those supercoils become even more compacted and form this type of shape that is referred to as the chromosome. So you essentially have like built into this structure here, these really supercoiled DNA structures. And again, with the accessibility for this information to be tapped into for later processes. So what is our mechanism for us to be able to look at or, um, tell that we have a DNA of interest? So a really simple but effective strategy is to take advantage of agarose gels to visualize DNA. So agarose gel electrophoresis is a method to determine the size of DNA, and it can provide you a bit of information with regards to the form of the DNA. So how does this process work? Essentially, DNA migrates through a highly cross-linked agarose matrix in response to an electric field. The phosphates on the DNA are negatively charged. So the molecule migrates to the positively charged electrode. So we basically take advantage of this charge and this applied voltage. Three factors affect the migration rate. One of them being the size of the DNA. And so as far as how we measure size based on the number of base pairs in the structure. Also, very importantly, the conformation of DNA. So as I mentioned, we can have linear DNA versus circular DNA. And another really important thing is the ionic strength of the running buffer. So what does that really mean? Well, it refers to the amount of salt in your buffer, and that amount of salt can affect the movement of the phosphates, which are highly charged. So here's just a cartoon representation to give us a depiction of how do we visualize the DNA. So in this agarose gel, what we have are four lanes, okay? And so in the first lane, the one to the left, what we have is a DNA ladder. So this is basically a ladder that is composed of DNA of different base pairs. And so what we see is how when you put the sample up here and you apply and you run the gel, okay, what you're going to do is move the nucleic acids that are in that ladder. So you have different ones of different sizes, or in this perspective, of different base pair amounts. And so the movement is such that the smaller DNA is going to move furthest, and the larger one moves slower. So essentially, you're forming this or I guess you can say this base pair ladder. So here's just a representation. This is a representation of 2,300, 1,800, 1,500, and so forth. The subsequent lanes can be, let's say, experimental samples, where, let's say, you isolated DNA from a tissue and you want to see what you've isolated in terms of like the size. And so you run them and you based on where they fall in terms of their movement through the gel, you then compare it to the DNA ladder to get a measure of how many base pairs compose that particular DNA. Okay. As far as the visualization process, well, DNA is typically visualized using a stain, aethylene bromide. And so this process of staining occurs because aethylene bromide, which is actually planar, it has the capability of intercalating into the DNA structure. And how is it intercalating? It's basically fitting within the base pairs. Remember, there's a stacked base pairs. So it fits within there, and then it provides this characteristic color. And so that's how then you're able to visualize the nucleic acids that are actually on the agarose gel. Another really important thing, I made a reference to this, is it also gives a little bit of perspective about the forms of a particular DNA that you're focusing on. So DNA conformation will affect the migration rate through the agarose gel. So we already mentioned how one thing that can affect the rate is how big in terms of how many base pairs the DNA is. So smaller goes faster, and bigger goes slower. Relaxed forms of DNA travel more slowly through the gel, with the circular form traveling slower than linear form. Okay, so that's important in terms of giving us a perspective of what are we going to see for, let's say, the same DNA itself in terms of how we visualize in the agarose gel. The supercore form will travel the fastest. Okay, so what we have here under this lane to the left is a visual depiction where we have some of the DNA that are basically multimers. So basically, for a reason, they are like forming more than one unit. But if we're focusing on just like the monomer, here we can distinguish them in terms of the type of structure that they might adopt, where you have the open circular form that moves the slowest, the linear form that then moves intermediate and in terms of its form, and then you have the supercore form that moves the fastest. And so you get a depiction of, what maybe a real type of experiment will look like for a particular DNA. Now, what you could do is you can take advantage of an enzymatic process for clipping this DNA so that you can simplify it into a linear form. Okay, so an example of that is a restriction enzyme that can cut the DNA and change it from these type of structures that are more compact into the linear form. And so when you apply this restriction enzyme, all of this more, I guess, complex behaviors that you see become simplified because now you've converted all that DNA into its linear form. And so in a perfectly executed restriction enzyme, what you will see is just one band that would correspond to the linear form of the DNA. So that's why this is labeled uncut versus the cut form. Okay. So that's just to give us a perspective of nucleic acids and tools that, or at least a simple tool that we can use for visualization purposes, and a bit about this the composition. So now what I want to move into is the genetic code and the principle behind DNA cloning and protein expression. Again, we're not going to go into all the nitty-gritty of it. We're just going to give an overview so that we're familiar with the process, so that's not foreign to us when subsequent lectures will refer to taking advantage of these process for, biological insight. All right, so this idea of the genetic code and the movement of information. So to the left, we have this depiction of DNA that undergoes a transcription, produces your mRNA transcripts, and then you have the process of translation that's going to take that information and produce proteins, or here's depicted as polypeptides. So the sequence of this protein, that order that we see here, is coming from the code of the arrangement of the nucleotides in the DNA. So we're going to just get a little bit of this idea of going from nucleotides to ultimately producing amino acids. This is what is referred to as the nucleotide code dictionary. And so here we have just a depiction of the sequence of the codes. And this is represented by three nucleotides defining a codon, and how the different codons on these nucleotides basically translate to a particular amino acid. And so you see some examples of how different arrangements of the nucleotides code for the same amino acid. So these are just examples of them. So again, more than one codon can specify an amino acid, except for methionine and tryptophan. Certain codons are also important because they are critical for protein expression. For instance, you can have this code AUG, okay, and that particular code can represent a start codon, okay. So if you look at this nucleotide code dictionary here, AUG also can represent methionine, so just keep that in mind. You also have certain codes that are representative of stop codon, for instance, UAA, UAG, and UGA. So this is important in terms of the arrangement of these nucleotides and how the transcription process can take place. So let's talk a little bit about then the workflow for amplifying a specific DNA gene and expressing protein from the gene. Right, so we're going to start from DNA, and then we're going to then talk about expression of proteins. So a bit about the workflow itself. All right, so how does this work? So let's say that you have a living organism and you want to extract DNA from them. So you can then take some protocol that allows you to isolate a tissue of interest from a living organism. Obviously, this will involve sacrificing the animal to then get the DNA. So this will probably be something that has a complex mixture that you then have to do some processing in order to try to get it to be more simplified, something that might be pure. Or you can start with buying DNA, plasma DNA, and that's then something that you get commercially. All right. So when it comes to then, trying to go from the DNA to then the process of getting an expressed protein, well, what you would do is from this DNA that's isolated from a living organism or from what you've purchased, here you can do a restriction enzyme to get access to a gene of interest, or you can go ahead and from that plasmid DNA that you obtained, do polymerase chain reaction, PCR, and then that can give you the gene of interest or even a modified gene. You can go from a gene that you have produced from an DNA that you've isolated, do a PCR experiment to then introduce a modification, giving you a modified gene. All right, so once you're at the gene level, then you take advantage of an enzyme called a ligase, which will then ligase that particular or introduce that particular gene into a cloning vector. And then once you have that vector that includes the gene of interest, then you can do what is referred to as cloning into a host cell. So the host cells can be different types of, it can be bacteria, yeast, so forth. Then in that process, sequence cells for the gene, you store the cells with the gene, or you transfer the gene into an expression vector. okay, to then move ahead with a protein expression, and that also will occur in a host cell. So you can stop at the gene level and then just store the gene of interest, or you can move ahead to the experiment where you want to go ahead to try to obtain your protein of interest and through the protein expression process. All right. Let's talk a bit about cloning in host cells. All right. So DNA cloning is the amplification of a particular gene or DNA segment by separating a specific gene or DNA segment from a larger chromosome, attaching the gene to a small molecule of carrier DNA, and then replicating this modified DNA millions of times through increase in cell number and the creation of multiple copies of the cloned DNA in each cell. All right, so this is basically the layout of what the DNA cloning process will entail. So what's critical to this process of cloning? Well, what's going to be critical are key enzymes that are going to do a number of different functions. Okay, so as an example, you have those enzymes that are restriction enzymes. Here, for instance, type 2 restriction endonucleases. What is the function? Well, the function of them is that they cleave DNA at a specific base sequence. And so we're going to look at, how that cleavage can occur. You also have enzymes that are referred to as a DNA ligase. And so, as the name implies, okay, you're basically ligating, you're joining two DNA molecules or fragments. And then you have the enzyme DNA polymerase, which facilitates the process of DNA replication. So, fills gaps and duplicates by a stepwise addition of nucleotides to the 3' ends. So, it's a successive building of this replication process. All right, so these are critical enzymes with their specific functions that facilitate the process of DNA cloning. All right, so 6 general protocols for DNA cloning. So they involve the following: cutting DNA at precise locations using sequence-specific endonucleases, which we will commonly refer to as restriction enzymes, right? So you know your DNA, and you want to use the restriction enzymes that are specific to where you want to have a cut. Then selecting a small molecule DNA called cloning vectors, which are capable of self-replication. Then covalently joining the DNA of interest, okay, so what you've cut initially, with the cloning vector using DNA ligase to form then your recombinant DNA. So that DNA is then going to be composed of your vector with the DNA of interest. Then transferring the recombinant DNA into a host cell, and then let the host produce multiple copies of that recombinant DNA. And then from that process, try to identify which of the host cells that have been now replicated contain the recombinant DNA that you're trying to produce. All right, so let's look at this with a visual depiction. You have your cloning vector. This is your eukaryotic chromosome that from which you want to isolate a particular gene. So the cloning vector is cleaved with the restriction enzyme. So this what you see here is like you have the circular vector. Then now because of the cut, you've now made a space into which you can insert your gene of interest. The same thing is done with the chromosome, where you will, select a restriction enzyme to cut it to get the gene that you want. So in this visual depiction, what is of interest is the red part. So you designed this in such a way that you cut on both sides, around that particular gene of interest, to be able to isolate that. So now you have your vector that's been opened up, and then you have your gene, the fragment that you want. So then you take advantage of the ligase to combine this particular fragment that's highlighted in red with the vector, and now you get your recombinant vector. So it's recombinant because now it's a hybrid between the original vector with the gene of interest. Then you have that recombinant vector and you want to introduce it into a host cell. So essentially a host cell will pick up that recombinant vector. And then essentially what you want to do is you want to propagate, you want to clone this several times so that you can produce a significant number of copies of your recombinant DNA. After that point, you will do a culture to grow to basically you're growing up these cells and you want to determine which of those cells contain the recombinant DNA for the purposes of storing them or using them for subsequent experiments. And now let's talk about DNA cloning to lead to protein expression. All right. So, modified DNA gene for the protein expression process. So, DNA cloning for protein expression requires modification of the gene using the nucleotide code for guidance. So, you're going to modify the gene of interest so that it includes recognition sequences, and those are critical to help facilitate this process. In the gene, for restriction enzymes that would enable the gene to be inserted into a cloning vector and expression vector. That's a critical thing. So include another recognition sequence in the gene that will enable the gene to be recognized by the expression machinery of the expression host cell. So that's another sequence that must be included. Also include nucleotide sequences that signal the initiation and termination of protein expression. Okay. So here's just a representation of a modified gene. This is a visual depiction of this. So you have here the 5' end to the 3' end. And so let's look at the components that we have here. So you have a restriction enzyme site. So that has a particular site that you want to have cleaved. The expression host recognition site. So you want that sequence to be there in order to then, be able to facilitate the expression process of, ultimately the protein in your host. And then you have the sequence that is the start codon, the actual gene of interest, the stop codon, and then another restriction enzyme that's going to be there. When you look at the complementary strand here, so this is referred to as the anti-strand, as this is arranged in the anti-parallel fashion. So the sense strand is 5' to 3'. This other is the complementary from the 3' to the 5' end. And that's, again, what we refer to as the anti-sense strand. Okay. So the sense strand of the gene, the 5' to 3' sequence, must have the appropriate sequences to facilitate cloning and protein expression. The sense strand, the coding strand, has the same sequence as the messenger RNA. The antisense strand, which will be the template strand, is a template for transcription. The polymerase chain reaction PCR can be used to create a modified gene. All right, so looking into then the choice of your restriction enzymes, your restriction endonucleases. So you cleave DNA phosphodiester bonds at specific sequences, common in bacteria, used by bacteria to eliminate infectious viral DNA. Some make staggered cuts. So that's important because they introduce what are referred to as sticky ends. Some make what are referred to as straight cuts or blunt ends. Okay, so we will see that in a moment visually what that looks like. And then there are a large number of restriction enzymes that are known and that are commercially available for us to use. All right, so here's a representation of different restriction enzymes. So as an example, BAM-HI, TAL1, and what's also very important is where exactly do they cut? And so that's going to be critical where you can see the cut here and it can cut over here. You also have those that cut in the same location. And so depending on how they cut, they introduce these cuts into a sequence, is whether they can form the sticky ends or you're basically going to have a blunt end. So the arrows indicate site where phosphodiester bonds are cleaved. The stars indicate site of methylation by DNA methylase. All right, so here's just a representation of a sequence. So this is coming from chromosomal DNA, and then like where we have certain sites that we want to cut. So this is a site for cleavage, the recognition sequence, and another cleavage. So according to what we have here, the sense of the antisense strand, well, we can use the EcoRI restriction enzyme. And what that's going to do is going to cut here and is going to cut here so that you're basically now are going to form these two ends. These are what we refer to as sticky ends, where you're going to be able to ligate something on them as long as you have a sequence that's complementary to the sequence that are present in these particular strands. To the right, we have a cleavage site where based on how the arrows are depicted, if you use this particular restriction enzyme, PVU2, okay, what that's going to do is basically going to form this blood end cut. And so that's a representation of how we can get sticky ends with one type of enzyme or a blunt end with another type of enzyme. All this is dependent on the code where exactly the nucleotide connections are. All right, so here we have just now this depiction of how we're going to take advantage of what has been cut by those restriction enzymes and then put them into a cloning vector. So you have this piece that has been generated with this sticky end over here. And so essentially you want to have your plasmid cloning vector to have a similar type of arrangement where you have a sticky end that's going to be complementary to this part of the sticky end of that sequence of interest. And then this blunt end needs to be complementary to what's over here. So you take advantage of a ligase to then fuse these that gene of interest into the cloning vector in order to generate this recombinant vector. So the base pairing complementary sticky ends greatly facilitates the ligation reaction. Blunt ends ligate, but they're less efficient in this process of ligation. Okay. So the ligase itself, so the enzyme that covalently joins two DNA fragments, normally functions in DNA repair. A human DNA ligase uses ATP as part of its process of ligation. Bacterial DNA ligase uses a nicotinamide adenine dinucleotide or NAD+. All right. A little bit about the cloning vectors. So the choice of vector to clone DNA depends on the size of DNA. So plasmids. So this is circular DNA molecules that are separate from the bacterial genomic DNA, can replicate autonomously. So you have origins of replication for use in bacteria and our yeast. They carry antibiotic resistance genes, and so we actually take advantage of this in terms of growing up, developing these DNA in certain organisms and using the antibiotic in order to restrict what type of growth we will observe, allows cloning of DNA up to 15,000 base pairs. To clone whole chromosomes, so here we're talking about going from 15,000 base pairs to 300,000 base pairs. So here you can take advantage of bacterial artificial chromosome BAC for use in bacteria, yeast artificial chromosome YAC for use in yeast, right? And that just gives us a depiction of the size of a gene of interest and then what vectors are accessible for you to try to take advantage of. All right, so here we have a visual depiction of a plasmid. So this plasmid is the PBr322, and then you have here the corresponding number of base pairs, so 4,361. All right, so what do we observe here? So what we observe here in terms of this we call the sequence are you have a part of it that has a ampicillin resistance. So what that means is that it's resistant to the antibiotic ampicillin. Over here, you have a sequence that corresponds to a tetracycline resistance, meaning that it's resistant towards the antibiotic, the antibiotic tetracycline. And so that's going to be important in terms of when you're trying to make your recombinant vector, and then you're trying to do the cloning process, what will actually grow. So a PBr322 has an origin of replication. So that was the site down here from this perspective of that plasmid. The sequence where replication is initiated by cellular enzymes. The plasmid contains two genes that confer resistance to different antibiotics. So as I mentioned to you already, tetracycline and ampicillin to allow this allows identification of the plasmid in host cells. And why is that? Well, antibiotics kill bacteria. Plasmids with certain antibiotics resistance allows growth, selection of bacteria that have taken up the plasmid on plates made with the antibiotics. All right, so let's take a moment here to process what does that really mean. So that means that this particular plasmid is designed so that you're basically taking advantage of bacteria as host, so that you, by building in what type of resistance it has, you can control what type of bacteria will grow. Okay, so having this built-in resistance, what type of antibiotics it's resistant to, will enable you to grow those, those bacteria with that particular vector, as opposed to bacteria that do not have that resistance will not be able to survive conditions in which they exist. Several unique recognition sequences, ECO-RI as an example, are targets for different restriction enzymes, endonucleases, providing sites where the plasmid can later be cut to insert foreign DNA. So those were, if you look back over here, sites where you have built-in, cuts that you can introduce. to put something of interest. The small size of the plasmid facilitates its entry into cells and the biochemical manipulation of the DNA. All right, so let's look at the transformation of the plasmid PBr322. That's our frame of reference. And so we were focusing on this. We were focusing on it having the ampicillin and tetracycline resistance. Okay, so let's imagine that we use a restriction enzyme and we're going to cleave this. All right, so we're cleaving this. And where do we cleave it? At least from this visual depiction here, this cartoon depiction, we're cleaving it at a spot where we had a sequence, a gene for ampicillin resistance. Okay, but this is a spot where we're going to insert our DNA of interest. So we take that foreign DNA, we apply ligase, and we now are going to ligase it into the plasmid. So the DNA fragments will be clonal ligated to the cleaved PBr322. Where ligation is successful, well, is in the location where we had the gene for ampicillin resistance. So because now you're inserting that foreign DNA there, you lose that ampicillin resistance. But what do you retain? So these particular plasmids, well, they retain their tetracycline resistance. So that's something you can take advantage of. Okay. So now you have this recombinant plasmid. It includes the DNA of interest. So what you're going to do is you're going to introduce them into the host. So E. coli cells are transformed, then grown on agar plates containing tetracycline to select for those that have taken up the plasmid. Okay, remember that this plasmid contains a tetracycline resistance, all right? So in theory, when you put this onto a plate that contains tetracycline, those bacteria that are going to grow are going to be those that are resistant to the effect, to the antibiotic effect of the tetracycline. All right, so that's essentially what we're trying to do. We're trying to insert this into the plasmid. So this basically you're transforming those cells to be able to receive these plasmids. All right, so the process by which recombinant DNA enters a host cell is called the transformation. One approach is by cooling the cells and DNA at 0 degrees Celsius, in a calcium chloride solution. Okay, so you cool it down, and then you do what is referred to as a heat shock. So you basically now go from 0 degrees to 42 degrees Celsius in a time frame of 5 minutes. So this is relatively fast. So that shock basically makes the host more permeable in order to insert that plasmid inside of them. Another approach is subjecting the cells and DNA to what is referred to as electroporation, which is you're going to apply a high voltage pulse, and that pulse does the same thing. It makes them more permeable and able to accept the plasmid of interest. All right, so identification of empty plasmids. So you're going to have a mixture, right, of hosts that contains the plasmid and hosts that do not. So you take this mixture, you put them on a plate, okay? And so this plate will have your antibiotic. In this particular case, the agar plate will consist of tetracycline. So what you will see as you plate this, you spread your mixture of bacteria, is that they start to colonize, okay? And when they colonize, they start forming these like aggregate structures. They tend to look white or off-white. And so each of these little colonies that you observe here, some bigger than others, well, in theory, they're growing because they have a resistance towards tetracycline. Other bacteria that don't have this built-in resistance, well, they won't grow, they won't form into these colonies. So in theory, these white colonies should have the plasmid of interest because they contain this resistance towards the tetracycline. All right. So then what do you do next? Well, we take advantage of the tools that are accessible. So let's say that you've pick up the colony and then you want to get access to the DNA that, the recombinant DNA that you've produced. Okay. So how do we do this? So we use a restriction enzyme to separate the gene from the vector because we know where cuts exist. So you can isolate your gene of interest. And then you run your DNA sample in our agarose gel because remember, that's our visual depiction of the DNA based on the size, based on their base pairs. So you see how your sample runs and where you visualize according to relative to the DNA ladder, you can tell whether you have a band that is of the size that you're interested in. that you're interested in. Okay, so let's imagine that that's something that we're interested in that has a base pair amount that's between 2,300 and 1,800, and that will signal that the relative success of this transformation process. Well, what's next? Well, you want to purify the DNA. So purify and sequence the DNA. The sequencing process is typically done by, or traditionally done by the Sanger method. I'm not going to go into this process, but it basically gives you the primary structure, the nucleotide arrangement of the DNA, and that would let you know where you're successful, in getting the DNA of interest. Okay. So now let's talk about the expression process. So expression of cloned genes. So to study the protein product of the gene, it's necessary to remove the gene from the cloning vector. Okay, so how do we go about doing this? You have special plasmids called expression vectors that contain sequences that allow transcription of the gene. The gene can be inserted with the appropriate digestion of the vector, and then ligase is applied. Expression vectors differ from cloning vectors by having a promoter sequence or promoter sequences, operator sequences, a code for ribosome binding site, because the ribosome is important for facilitating the translation process, the transcription termination sequence. That's part of this as well. All right, so let's look at a representation of a typical expression vector so we can see the components and some of the things that we just mentioned. All right. So you have the ORI site here. You have a site for the ribosome binding site that's going to facilitate the translation. These bacteria promoter and operator sequences here. You have a site where you want to introduce your gene. So when you think about how you're going to introduce your gene, you have to think about the sequences that are here and what type of restriction enzyme will facilitate you to insert your gene of interest. You have your transcription termination sequence and then your selectable genetic marker which can have in certain built-in, antibiotic resistance. So these are some of the critical components for this expression vector. Okay, so what's really important is that the vector needs to be specific to the host cell. Okay, so expression from bacterial cells typically done to obtain a non-glycosylated mammalian protein. So this is actually important because certain organisms that are used for expression purposes may modify your protein of interest in different ways. When typical proteins may be glycosylated, so may contain some sugar molecules or may not be. So it depends on what you're trying to produce in terms of what the protein of interest is, whether you want the glycosylated form or not. So normally one takes advantage of bacteria because bacteria, in terms of the machinery, will produce the non-glycosylated version of the protein. All right, so then if you want to express in bacteria, it's again critical that the expression vector is something that can be used by that particular host cell. The promoter allows efficient transcription of the inserted gene, and the operator permits regulation. Okay. So the ribosome binding site provides sequence signals for the efficient translation of mRNA derived from the gene. The gene to be expressed must include a sequence specific to the host cell, just as I was just mentioning, I probably should have put this earlier, which is important for ribosomal binding. So, for instance, expression in bacteria, you want the Shine-Dalgarno sequence, which consists of this particular sequence. For eukaryotes, the Kozak sequence, which consists of this sequence that we have here. Okay. Again, choice of vector needs to match the choice of host cell. Okay, so let's talk a little bit about creating the modified gene. Okay, so here we're going to invoke PCR, which is used to amplify DNA in the test tube. So you can amplify regions of interest genes within linear DNA, and you can amplify complete circular plasmids. So the mixture for PCR will consist of the following. Will consist of the target DNA, primers, which are these oligonucleotides that are complementary to the target DNA, and then you can also introduce nucleotides because if you're trying to like replicate the DNA, you're going to need the nucleotides that are going to help facilitate this replication process. So we include these nucleotides and also thermostable DNA polymerase to facilitate the actual polymerase chain reaction process. Okay, so you place the mixture into a thermocycler. So that's something that's going to facilitate controlled changes in temperature. So what is this process that you're controlling? First, you're going to introduce a 95-degree round where that's a very high temperature. What that's going to do is going to melt your DNA. Then, in this process, you're going to have separated strands. So here, then, you cool the separated strands to about 50 to 60 degrees Celsius. Then you have primers anneal to the target. Then you use a polymerase to extend the primers in the direction of 5' to 3'. And then after the round of elongation is done, you repeat these steps. Okay, so let's look at this from our perspective here. So this is the region of target DNA to be amplified. So you do that heating process, okay? And so that heating process is going to melt, is going to help to facilitate the separation of the strands. You also add in the synthetic oligonucleotide because that's what's going to help you to extend um, the um, the regions of interest, once these strands are separated. So here's just that depiction of the separated strands. You add the thermostable Taq DNA polymerase to catalyze then the extension 5' to 3' for DNA synthesis. So you have the 5' end, you have this polymerase, you have the nucleotides that you introduced there, and you basically want to base pair match the sequence you have here, the 3' and 5', the 5' with 3' end. Okay. And so you're matching that with the nucleotides using the polymerase. And so that's how you get this extension. And that's essentially the process of replication. Okay. So you do this extension. So then you basically repeat steps 1 and 2, which is you have now the replicant strand. You will heat up, separate the strands, then use the polymerase to then extend along with the primers and obviously the nucleotides that are facilitating these extensions. Okay, so then the DNA synthesis is catalyzed by that polymerase. So you do multiple rounds of this. Okay, and you repeat this until after 20 cycles, the target sequence has been amplified about 10 to the 6th fold. So many orders of magnitude. You've now replicated this region of interest. Yeah. So creating the primers for a desired gene. Okay, so that's going to be crucial. The primers are what's going to facilitate how you extend, like you're base matching the strands, and you want to make this decision carefully so that you don't have a mismatch or you have an offset in terms of what you're trying to replicate, which can happen if you're sloppy with the design of your primers. So say you want to clone a gene using a vector with an E-coli cut site and want to express protein in a eukaryotic cell also with an E-coli cut site. The gene will have to be modified by performing PCR with the following primers. So we're trying to introduce in the protein um, this particular cut. So for us to do so, we will use a primer that includes this particular sequence here. So we have a 5' GAA TTC Kozak sequence ATC ATG subsequent nucleotides 3'. So we're basically going to have this 4 primer that's going to facilitate what we're trying to introduce into the protein that we're going to express. And then we also need the reverse primer that essentially is going to look somewhat of this nature. So those areas that are marked XXX, these are basically consist of the nucleotides that are part of this primer. It could be 18 to 20 nucleotides. For the reverse primer, basically it's a complementary strand. So that's we have XC because as it matches up, it will be complementary to that sequence. Okay. So recombinantly expressed proteins will have a methionine at the end terminus. So the typical start codon, I mentioned this before, I think I just said the AUG, so ATG or AUG codes for methionine, and consequently, that amino acid will be part of the expressed protein sequence. So let's imagine that that particular amino acid is not something of interest that you want in your protein. Well, then you can have that N-terminal methionine removed by the application of a methionine aminopeptidase. So that's just going to cleave the methionine from the end of that sequence. And then you'll get the protein with the sequence that you actually want. All right, so in terms of being able to use this to our advantage, so what you want to do is, let's say, to introduce mutations because you want to get access to mutant proteins to look at the activity of certain mutations as compared to the wild type protein, or you want to introduce certain mutations that can help facilitate the process of purification in a manner that's much more easy for you to do. So this is some of the reasons why you introduce gene mutations. So alterations in the clone genes, so what that does, it will produce modifications in the proteins that are going to be expressed. So cloning techniques can be used to produce protein products altered from their native forms. So you can have what is referred to as a site-directed mutagenesis. That is, a DNA segment can be removed and replaced with a synthetic one that is identical to the original, except that you introduce a desired change, a desired change. So a desired change in the coding is going to give you in your final protein um, a different amino acid that would normally have been part of that sequence. You can also do what's referred to as an oligonucleotide directed mutagenesis, where a specific DNA sequence change can be performed. So you have more than one site that's being modified. All right, so this is just a representation of this. You have your site-directed mutagenesis. Here's your recombinant vector. You cleave it with a restriction enzyme. Here you have now opened up that vector. And so you're taking that part of the gene, you're removing it, and now you're introducing a mutant form of it that has, let's say, one site that is going to be mutated, is going to give you a different amino acid that's going to be when you do the expression. So you insert that into um, this vector, and now you have this mutant form of it. Here, as I mentioned to you, the oligonucleotide-directed mutagenesis. So you have this gene target site for mutation. So essentially what you want to do is you will try to modify that gene. You introduce primers, so denature and anneal oligonucleotide primers with mutation. You use DNA polymerase to extend and incorporate the mutagenic primers. Then you digest non-mutated parental DNA template with methylation-specific nuclease and anneal newly synthesized strands, so mutated plasmid nicked strands. And then you transform the DNA into cells, and the cell repair nicks in mutated plasmid. So that essentially is what the nucleotide directed mutagenesis is. All right, so really critical. So let's say you're trying to introduce a mutation that you want to study, or and this is something that is used often in biochemistry, you want to facilitate the process of isolating your protein of interest. And so what you introduce is a tag, because you want to take advantage of the properties of that tag to help with purification of the protein, because protein purification can be quite complicated. Because in addition to expressing proteins, if let's say you don't use a tag, you not only are you expressing your protein of interest, but the host machinery is expressing other proteins. So you're going to get a complex mixture. And so to purify this complex mixture can be, quite intense. So that's why people will try to introduce a tag to distinguish their protein of interest from what's being produced by the host on itself. And then that will help to be able to get access to that protein by the purification process. So purification of natural proteins is difficult for just the fact that you have just a mixture of many different things, and the tools that are available can help to simplify, but you might have to use several dimensions of purifications, and that may not be easy in terms of the time that is involved and also the corresponding funds that are involved. So recombinant proteins can be tagged with specific amino acid sequences for purification by inserting the gene encoding the protein in a vector encoding the amino acid sequence tag at either end of the gene. So let's say you put it at the what would be the 5' end or the 3' end. And so with respect to the protein, that would be the N terminus or the C terminus. The protein is then purified by some sort of affinity purification. So an affinity for the tag that you have introduced. And so by the tag being there and being binding to a solid resin that has an attraction from it, all the other proteins that have been produced by the host that do not have this tag and thus have no affinity for the resin will just wash through. And then what you need to do is an elution process to basically disrupt the affinity of your tag protein from the resin that has an attraction for it. And then you get access to your isolated protein. So these are some examples of common tags that are used. One of the major ones is a histidine tag. And the histidine tag can consist of different numbers of histidines, but typically consists of 6 histidines in a row. And so you can immobilize it on a resin that consists of the metal nickel, because nickel, in terms of its metal properties, has an attraction for um, molecules like histidine. So that requires a little bit of like metal chemistry, but just the understanding that the nickel is attracted to the histidine, and that binding of the histidine to the nickel is what then allows you to separate your protein of interest. So here you have other examples of tags. So protein A and the corresponding ligand for that particular protein. Um, so just a representation of what you can use and what type of um, resin with an immobilized ligand that you can take advantage of as a binding partner for that tag. But also very important to keep note of is the size of these tags. They can vary greatly. Histidine tag is relatively small, zero point 0.8 kilodaltons. Some of these other ones are quite big, okay? So they can potentially introduce um, structurally a change in your protein. And so in addition to, helping you to purify, you also have to think about, do I need to get rid of this tag because it's going to alter the protein that I'm interested in? And even something as small as a histidine tag, you might want to consider that you might need to actually get rid of it after you've done the purification process. Sometimes histidine tags are pretty innocent, so they don't affect anything about the protein, its structure, its activity, but sometimes they can. Okay, so these are things to think about with the use of tags. And then downstream, you've obtained your protein of interest. Um, are you good enough for what you want to use that protein? So this is just an example, where we introduce a tag. We have a vector that includes a tag of interest. Here I'm just showing you a histidine tag. And so we have a DNA encoding protein. You ligate that into that vector. So by that ligation process, you have your gene that will ultimately be used to produce a protein, but then is ligated to that histidine tag so that when this is expressed, you're going to express your protein plus the tag. And then here's just a representation of, you have your sample, which is going to consist of your lysate. So your lysate is just like the homogenized um, cells if you're doing bacteria, for instance. And so you're going to get your proteins of your protein mixture, and so you put it through a column. So here's just a little cartoon representation of a column. So if you have a histidine tag, your column should consist of nickel, because nickel has an affinity for histidine. So here's just a representation of a nickel resin where you have nickel sites that are accessible for binding. And that is what the histidine tag takes advantage of. Histidine molecules, this imidazole group, will bind to the nickel, and that binding interactions will then facilitate the purification process. So you bind, you wash. What does not have an attraction for the nickel will wash through, and then what binds to the nickel will stay stuck, and then you have to do some sort of elution process to then remove the protein with the tag to then isolate your protein. And that's just an example of the purification process when you have a tag present. All right, so with that, I actually got through the slides, but that's a lot of information. Um, just wanted to review these things with you. I have recorded this lecture, so just in case you want to review this, I've already posted the slides, so you can look it over. But yeah, so thank you. And so I will provide you the recording will corresponds to your project, and then we'll resume as already depicted in the course syllabus. If you have any questions, doubts, just let me know. You can contact me. All right, everyone, have a good rest of your day, and I'll see you next week.